All right, welcome back. So this is probably going to be our last part for this eye lecture. And so what we'll finish here is talking about receptive fields. We'll go into a little more detail about the actual phototransduction cascade, and we'll look at some more images to hopefully make a little bit more sense to you. And then we'll look at the actual pathway of the visual stimulus leaving the eye to the optic nerve and how it actually travels in the brain, and then some clinical kind of correlates related to what can happen if you damage a portion of this pathway. Okay, so talking about receptive fields here. So if you recall from a general nervous system, general sensation lecture that we did previously, chapter 12, we looked at the receptive field for a neuron, right? So the receptive field was one neuron with its one axon and all of the sensory receptors, all right? Well, the receptive field for our eye is essentially the same thing. It's the ganglion cell and then all of the area of the retina that captures light that goes to that one ganglion cell. So all the cells involved in the capture of a single photon, so one unit of light, including the ganglion cell that signal converges on. Okay, so if you look here, there's one ganglion cell down here. So if we're looking at this inferior part, there is one single ganglion cell that we see right here, and it is receiving information from this photoreceptor. It's receiving information from maybe this photoreceptor, and it's receiving information from this photoreceptor. So this is this convergence, and we've talked about converging circuits. This convergence from all of these kind of presynaptic neurons, these photoreceptors, to a bipolar cell down to a ganglion cell. All right. So what do we have are what are known as on-center and off-center neurons, or on-center and off-center ganglion cells. Okay. So what this means is that certain ganglion cells are stimulated or turned on when light strikes the center of their receptive field. So if we look at this here, and the receptive field for these neurons is usually circular. So the receptive field is usually kind of circular in a circle-like structure or organization. So what we see up here is we see this structure. So we see the center here kind of shown in yellow, and we see the surrounding area all in blue. So if this neuron down here, this bipolar cell, is an on-center, then it is stimulated to be turned on. So you see this positive. It is stimulated when light strikes the center of this receptive field. All right. If this was an off-center, so we look at this one right here. We're looking at off-center. Then it is inhibited when light strikes the center, but it is stimulated when light strikes the periphery. So you see all this outside. We are stimulated when light strikes here, but we are inhibited when light strikes in the middle. So this. And again, this is for just one receptive field, and all throughout the eye we have hundreds and thousands of these receptive fields. And so when they all converge, so this one on-center ganglion cell is turned on here, but you may have some off-centers somewhere else, and more on-centers and more off-centers. Whenever these are all turned on, the on-center and off-center, and they all kind of converge and mesh together in the signal sent to the brain, this gives us very good contrast and visual acuity. All right. So things like edge detection. Whenever you're able to tell the fine edges of a structure or of an object, or the specific details and the layout of it and the actual form of it, this is accomplished because of these different ganglion cells that are turned on in different patterns, right? Some are turned on when light strikes the center. Some are turned on when light strikes the edge. Some are turned off when light strikes the edge. Some are turned off when light strikes the center, right? And so this contrast between these two, and this kind of combination of on-center cells turning on and off-center cells turning on, then you get this nice sharp visual acuity, this nice contrast in terms of the edge of being able to determine the edge of structure. So that's known as edge detection. Okay, so this is a video. We're not going to watch it in this video. So if we look closely at our phototransduction cascade, this is what we're going to look at. We're going to look specifically at rods. Okay, but the process is more or less the same for our cones as well. So what we have is we have our rods in a kind of a resting state. Okay, so whenever there is no light, they are turned on. Okay, you can think of them as being on. All right, they are packed with these proteins, or these photopigments, so this rhodopsin. And so the rhodopsin is a combination of opsin and retinal. Okay, so opsin is a little protein, and retinal is this structure you see in the middle. So opsin is this large kind of protein we see out here, and retinal is this kind of convoluted structure we see circled here in red. Okay, So whenever we stimulate our pigment, so when we stimulate this rhodopsin with light, we actually convert this retinal protein into a different form. This conversion into a different form further leads to opening or to actually closing of ion channels, which results in hyperpolarization, which releases less glutamate, and we hyperpolarize, and then we stimulate our bipolar cells. Okay. So again, like we mentioned in the previous part, it's somewhat counterintuitive in terms of what happens when light strikes. So when the light strikes, it actually turns this cell. You can think of turning this cell off, if you will. Okay. So if we look at this example here, if we start here with 1, so we have light coming in. All right. So this rhodopsin is absorbing a light ray. Normally, this retinal pigment here is in what is called the cis form. Okay, cis form means that it is in this bent form. So it comes down and then it bends down like this. All right. After light stimulates it, it is converted into what is known as the all-trans form. Okay, and this trans form means it is more straight. Now, so we went from this kind of bent form to this straight form. All right, this bending or this straightening out of the retinal causes, and we'll see this in the next slide, causes this pigment to then bind to a protein known as transducin. Transducin activates another protein called phosphodiesterase, or PDE, and then phosphodiesterase ultimately reduces the intracellular concentration of a molecule known as cyclic GMP, and this closes sodium channels. And so when sodium channels close, you then hyperpolarize the cell. You get fewer graded potentials, so you get less glutamate release, so you get stimulation of the bipolar cell. Okay, so we've mentioned this a few times here. All right. This is then showing how we can revert back into, we can restore this retinal and opsin complex here, and this is a process known as bleaching. Okay, so for bleaching, you actually have to split the retinal and the opsin apart. So here you see they're together still. There's an opsin, there's a retinal here. You actually have to break them apart. So retinal to one side, the all trans, and the opsin to its own part, and then you have to use ATP. So we talked about previously in that inner segment of the rods and cones, you have mitochondria. You need mitochondria because you need to make ATP, because ATP is going to configure this retinal into its cis form, so this bent form, and then it can put it back, and then you get your restored rhodopsin. So this process here is known as bleaching. Okay, this process is known as bleaching. All right, so if we look at this picture, hopefully this will make this process look a little bit more sense. So again, we're looking at a rod here. Okay, we're looking at a rod because we're dealing with the protein rhodopsin right here. All right, so we're looking at a rod. So what you have first, you have light come in. So you have light from, let's change colors. Let's make light, we'll make it orange. Okay, that's more light. So we have light coming in through our cell. It comes all the way in, and it strikes our rhodopsin protein, and it hits that retinal. Okay, and it changes that retinal into a different form. Okay, now note, let's move over here for a second. We have these channels, so let me switch back to red here. We have these channels over here, all right? These channels are sodium channels, and normally, while this cell is on, these sodium channels are open, okay? The opening of sodium channels allows sodium to come in. This depolarizes the cell, and it sends a signal down the cell to release glutamate, which inhibits the next cell, which inhibits that bipolar cell, okay? So after we have the light come in, and we strike this rhodopsin, and we convert the retinal from cis to all trans retinal, we activate a protein called transducin, okay? We activate a protein called transducin. The transducin binds a molecule and then comes over here and activates this protein here called phosphodiesterase, okay, PDE, phosphodiesterase. Phosphodiesterase takes this cyclic GMP, so these molecules over here in purple are known as cyclic GMP. They are required to keep the sodium channels open. So if they're a cyclic GMP, you keep the sodium channel open, right? Whenever you activate this phosphodiesterase protein, you take that cyclic GMP and you convert it into GMP. Okay, so you take the cyclic GMP and you convert it to GMP. Well, when you convert cyclic GMP to GMP, you've now reduced the amount of cyclic GMP. So that causes these protein channels to close. So when they close, you have less sodium coming in. So instead of depolarization, you now have hyperpolarization, and that turns the cell on. Okay, so now the cell is on. So the cell is said to be hyperpolarized. Now that the cell is hyperpolarized, it's going to release less glutamate at its synapse with the bipolar cell. So if there's less glutamate, then you actually stimulate the bipolar cell, and so the bipolar cell knows that a photon of light has been received, right? So what we mentioned right here, it received a photon of light. That then stimulates the ganglion cell, and then the ganglion cell sends a signal back to the brain, and we interpret this as some type of vision, right? So this is referred to as the phototransduction cascade, right? Cascade because you have one event happening here with light and the retinal being changing forms. You have the activation of this phosphodiesterase, which changes cyclic GMP to GMP. When you convert this to GMP, you close these channels. When you close the channels, you prevent sodium from coming in, so the cell hyperpolarizes, so there's less glutamate release, so you actually activate the bipolar cells, okay? So when all of this happens, whenever we have rods stimulated, when we have cones stimulated, we have different types of vision that results from this. So we have one type of vision called scotopic vision, all right? And scotopic vision is a result whenever we have rods be stimulated, okay? This is generally vision that occurs in low light environments, right? So we have low light, and we said previously that when we have rods, these are not very good at color vision, but we can see general shades of gray and black, okay? You have poor edge detection, like we mentioned previously, but you can see general shapes of things, but not very clearly, okay? Now, when we have our cones activated, we have something known as photopic vision, okay? So photopic vision is generally happening and occurring when we have lots of light. So we have very bright light conditions, very intense light, and this is what allows us to be able to see very distinct color, okay? So you may remember previously we talked about that we have different types of cones. So we have mostly red cones, we have a few blue cones, and then even fewer green cones. And remember, these are responding to various wavelengths of light, and the stimulation of certain blue cones and certain green cones and certain red cones gives us all of the colors of our visual spectrum, right? So red, orange, yellow, green, blue, indigo, and violet, and kind of all those shades in between, okay? All right, so this is showing, again, what's happening in the dark versus what is happening in the light, and we're looking at rods here. Okay, so this is basically the same thing that we were showing earlier. So in the dark, we said that this is going to be the cell is essentially going to be turned on. Okay, so you have sodium coming in, right? You have sodium and influx in, potassium efflux. You have glutamate being released down here at this synapse, and when glutamate is released here, remember that glutamate is inhibitory. It is an inhibitory neurotransmitter, so it is negative. So that inhibits this bipolar cell, which is this kind of pink red one down here. And when that happens, we do not have any action potentials be sent to the ganglion cells, right? So if we look at this graph here to the right, we see exactly the same thing. So whenever we're in this dark current, so in the dark, no light, we have sodium coming in, and if you recall from our action potential lectures, the entry of sodium causes depolarization when one reaches threshold, right? And so our photoreceptors don't generate action potentials, but they generate graded potentials, right? So if we're looking right here, they can generate graded potentials. So you can get this graded potential traveling down the axon, right? This graded potential is excitatory because we're depolarizing the cell, right? So you have calcium channels open, just like we had in our action potential. Neurotransmitter is released, and in this case, it is glutamate, right? So we're releasing glutamate here at the synapse. The binding of glutamate hyperpolarizes the bipolar cell, and that is inhibitory. So this bipolar cell here is inhibited. Well, if it is inhibited, it can't send a signal to this ganglion cell down here. So the ganglion cell is also going to be inhibited, and there's going to be no action potentials. All right. So the binding of glutamate hyperpolarizes the bipolar cell, which causes inhibition. There's no release of glutamate at the bipolar cell from the bipolar cell onto the ganglion cell. So you have no nerve signals being generated by the ganglion cell. So you would have no signal going to the brain. All right. So again, this is the dark current. This is what is happening in the dark, right? So in the absence of any light. All right. So if we now have light, then we get basically the exact opposite thing, right? So when we have light now, we've converted that retinal from cis to trans. We now have a lack of sodium coming in because we closed the sodium channels, right? And so the cell hyperpolarizes, right? So if you look here, we are hyperpolarizing the cell. We have fewer graded potentials traveling down here, down this axon. So you have less or no glutamate release. So if you look here at the synapse, no glutamate release. So if you have no glutamate release, that's actually excitatory. It is actually positive for this bipolar cell, right? So it's excitatory. So right here, it's going to depolarize. When it comes down, it can release glutamate at this synapse. And at this synapse, glutamate is actually excitatory. So whenever we release glutamate here, that's going to be positive for our ganglion cell, and that's actually going to send a signal back to the brain, to the occipital lobe, and we're going to perceive that as some type of light, right? So the end result here is we're actually exciting our ganglion cells because we initially hyperpolarized our photoreceptor, okay? With the previous one, we depolarized the photoreceptor, so we inhibited the ganglion cell, okay? So keep these things in mind. Keep in mind sodium coming in, the graded potentials traveling down, the release of glutamate, and hopefully this will begin to make sense regarding what happens when you have light present, what happens when you have no light present. Okay, so this is kind of the pathway that light travels through our eye to reach our retina. So we have the light pass through our cornea. So if we're looking here, light is going to come through, strike our cornea. It's going to go through our pupil into that anterior chamber of the eye, in the anterior cavity. We're going to go through the pupil, so through that dark part in our eye, through that opening, to the posterior chamber. Remember, that's still part of the anterior cavity. Through the lens. Remember, the lens is this kind of biconvex structure that is then going to refract light and focus it onto our fovea, right? Our fovea centralis. This posterior region of the eye back here, this is that vitreous cavity or that posterior cavity. We're then going to have our retina, right? So this posterior layer way back here goes through our vascular tunic. It's going to go through all these layers. And then we're going to have a signal, and you can see it in this image to the right, be sent along what is called the optic tract, optic nerve, and optic chiasm, these different structures, and we'll look at this, ultimately back to our occipital lobe of the brain, where we then perceive this as some type of vision, okay, or some type of image. Okay, so we have photoreceptors, we have our bipolar cells, we have ganglion cells, and then the ganglion cells form a bundle, this kind of aggregation of their axons going to the optic nerve, right? So remember, light has to go through, strike the pigmented epithelium, and then we can have the signal go back the opposite way and then down toward the brain. Okay. All right. So when we look here, we have a specific pathway of the action potential, and this is assuming we have now stimulated our rods or our cones, and we are sending a signal back to our brain. All right. So first off, we have our optic nerve, and we've mentioned the optic nerve a few times. This is the structure that is leaving the eye at the optic disc. So where the retina converges becomes the optic disc, and then it's leaving as our optic nerve. Okay, so this is our second cranial nerve. So cranial nerve 2. All right, so I'm going to write cranial nerve 2. All right, this is our optic nerve. Okay, so this is what we see right here in red. Okay, this is our optic nerve. Now, I'm going to change colors here. Where the optic nerves meet, so we have a right optic nerve and we have a left optic nerve, where these meet is a region known as the optic chiasm. Okay, so what is the Greek letter chi? What letter is that? So the Greek letter chi is this right here. It's X. Okay, so we have this X structure running right in the middle here. Okay, so that forms this X in this middle region known as the optic chiasm. Right. Now you have fibers cross at the optic chiasm from both sides. So you have the right eye and the left eye being crossed here, fibers from both right and left optic nerves crossing at the optic chiasm. But you also have fibers from the right and left optic nerve staying on the same side of the brain. Okay, so if I change colors one more time, let me do something like green. So you have some fibers coming from here that cross over, but you also have some fibers that stay on the same side. Okay, and we'll switch colors again. Let me do something like yellow. You have fibers here crossing at the chiasm, but you also have some fibers here staying on the same side. All right, so it is depending on where the fibers originate from along the retina that determines whether or not they cross to the opposite side of the brain or they stay on the same side. And we'll look at this. I'll show you in the next slide. I'll kind of lay this out for you a little bit more clearly. Okay, but from this, we went from the optic nerve, right? So I'm going to show here. This is the optic nerve. This region here is known as the optic chiasm, and this region back here, this is known as the optic tract. Okay, so our optic tract here, right? It then goes to the thalamus, to a region known as the lateral geniculate nucleus, and then from there, fibers project to our occipital lobe of the brain, where we have our primary visual cortex, and this is where we have interpretation of the information that is coming from the environment, stimulating our retina and traveling back to the brain. Okay, so let's take a look here at this region of our optic nerve here. So what we have is we have signals, so action potentials ultimately, coming from both optic nerves that are going to both sides of the brain. So let's look at this image up here to the left first. All right. So I'm going to show this in green for nice contrast. So say we're looking at this eye here. Say we're looking at this eye right here. If light is coming from the medial aspect of our field of vision, so this region in here, this is the medial aspect, right? If light is coming from here, it is going to stimulate the lateral part of our retina. Okay, this is sometimes called the temporal retina. So if we look here, here's our temporal retina because it's closer to the outside. It's closer to our temporal region, right? So here's our temporal region over here. Okay, so as light comes from the medial aspect, it strikes the temporal retina. And the fibers here come down the optic nerve to the optic chiasm, and they stay on the same side of the brain. Okay, that's important. So signals coming from the temporal retina stay on the same side of the brain. We see the same thing here on this right image. We're coming from our medial field of vision. Our medial field of vision strikes the temporal retina. We come along the optic nerve to the chiasm, and we stay on the same side of the brain. Okay. However, if light is coming from our lateral field of vision, okay, so say it's coming from, if we look here, if we're coming from way out here, so say we now have light coming in, it's going to strike what is known as our nasal retina, okay? Nasal retina meaning the side of the retina that is closest to the nose. It's more medially located, okay? So that's this region on this image to the right. That's this region right here, okay? So if light is striking our medial or our nasal retina, that information, those action potentials, will go along the optic nerve to the optic chiasm, and they'll actually cross to the opposite side of the brain. So right here we see that information from one eye is going to the same side of the brain, and it's also going to the opposite side of the brain. So if we look here at this example on the right, I'm going to clear this just to make it a little bit cleaner. Here we have light coming from our lateral field of vision, striking our nasal retina, right? Signals are then going to come along the optic nerve to the optic chiasm, and then they're going to cross to the opposite side of the brain, right? Again, if it was coming from the medial field of vision, it's going to strike our temporal retina right here. Information is going to go along the optic nerve toward the optic chiasm, but then it's going to stay on the same side of the brain, okay? So, and this occurs for both eyes. So I'm just going to show you in one eye, but the process would be true for this eye here, right? If we came from our lateral field of vision, we struck our nasal retina. We're going to then come along our optic nerve. We're going to come to the optic chiasm, and then we're going to go over to the opposite optic tract to the opposite side of the brain. Okay, so the pathway of nerve fibers is really from the retina to the optic nerve to the optic chiasm into the tract. Now, not every fiber is going to cross the chiasm, but it may approach it and then, so right here, so if it's coming here, it may approach the chiasm and then stay on the same side of the brain. All right. So what this means is that if we damage either our optic nerve, our optic chiasm, or our optic tract, then we can have certain deficits, right? So I'm going to switch to this black color here, okay? Because black, this is a negative thing, right? We're having a disease or some pathology happen here. All right. So say we have damage to our optic nerve. Okay, so we're going to look at this image to the left. Say we damage this optic nerve right here. We cut it. Okay, so that's what this line means. That means this structure is damaged. All right. What type of visual deficit would you see? And for all of these, we're going to assume complete damage to whatever structure. Okay, so complete damage to the optic nerve, right? So if we damage just our left optic nerve, what would happen? Well, hopefully you're saying we would lose vision in our left eye, and that's exactly right, right? So if we lost it, information coming from this nasal retina and information coming from this temporal retina, both of it is lost because we've lost this optic nerve, so none of the information can reach the brain, right? So information is not going to go to the chiasm and cross, and it's not going to stay on the same side, okay? So say we damaged our optic chiasm, okay? So say I come in here and I put a big X right here. We've damaged our optic chiasm, okay? Well, what type of visual deficits are we going to see? So to understand what visual deficits we're going to see, just trace back where the fibers are coming from. Well, what fibers cross at the chiasm? Well, it's fibers coming from this nasal retina, right? So that means vision coming from this lateral visual field. So vision coming from out here strikes this part of the retina, it then stays in the optic nerve, and it crosses at the chiasm. Well, we damaged our chiasm, so we have lost that vision. So in other words, we've lost peripheral vision, okay? We have lost peripheral vision from the outside. Same thing here. We have vision coming from the periphery, strikes the nasal retina, comes along the optic nerve to the chiasm, but it's damaged, so it cannot cross, okay? So if you have a total loss, total severing, total damage to the optic chiasm, you have lost your peripheral vision. So you have what you could think of as tunnel vision, right? You can only see things in your medial visual field. You cannot see things out to the periphery. Okay, so obviously very dangerous. Okay. All right, last one. Say we damage our optic tract. So say we damage this structure right here. Okay, so there was some type of lesion, something happened, and we damaged it. All right. Well, what visual deficits are we going to see? Okay, well, same thing. Let's trace the fibers back. Let's trace these orange ones back. Okay. So I'm going to change colors here and do this. So let's trace these orange ones back. So we're going to trace it here. All right, so we've lost this temporal retina, which means we've lost information from this medial visual field. So we've lost vision from this part of our visual field. All right, let's trace these blue fibers back here. All right, we crossed at the chiasm to the nerve. All right, now we're at this nasal retina here on the opposite eye, and we've lost from this visual field over here. All right. So what this is we've lost the same side of our visual field in both eyes. Okay, so say we were looking down on this. So this would be the left eye here. This would be the right eye here. Okay, so we've lost the right side of our left eye, or in terms of the visual field, the right visual field in our left eye, and we've lost the right visual field in our right eye. Okay. So you've lost the same side of your visual field in both eyes, right? If we damage this optic tract, then it would be exactly the opposite. But we've damaged the right visual field of the left eye and the right visual field of the right eye. So what this is called is homogeneous hemianopsia. Okay, so this really very complicated, strange term, homogeneous hemianopsia, meaning we've lost the same homogeneous, hemi, meaning the same side, one side of our vision. Okay, so homogeneous hemianopsia, we have lost the same side. We've lost the right side, the right visual field of our left eye, and we have lost the right visual field of our right eye. Okay, so say we lost the opposite tract, right? So say, let's trace this one out just for example's sake. Say we damage this one here, right? We damage this one. Well, if we trace the fibers out, we should get the same thing, but on the opposite side. So here we've lost these orange ones here. So we've lost this visual field of our right eye. So again, remember this is right and this is left. And if we trace these blue ones out here, we've lost this nasal retina, so we've lost this visual field over here, okay? So here we've lost the left side of the visual field of the right eye and the left visual field of the left eye, okay? So medial vision from the right eye, lateral visual field from the left eye, okay? But it's the same. It's the left side of the visual field in both eyes. And so again, this would be homogeneous hemianopsy, all right? So not every time you damage, so say not every time you damage the optic nerve, the optic chiasm, or the optic tract, you don't always lose the entire thing. So you don't always damage the entire optic nerve, the entire optic chiasm, or the entire optic tract. But for example's sake, what we're talking about here, we're saying that we do, it just makes it a little bit easier to learn the symptoms, what visual deficits you would see, okay? So here you can see on the right, here's this visual field from here. Here's the visual field from the opposite eye here, all right? So visual pathways, we have both eyes converging on similar fields. So here we have visual field from the right eye. So this would be as if we were looking on the underside of the brain, okay? So this is the underside. So this is the right side over here. This is the left side over here, all right? That's a bad L, sorry. And then here we have the left eye only. So where we cross, where we have visual fields being picked up from both eyes, this is our binocular vision, right? So binocular meaning coming from both eyes, all right? And this allows us to have our wonderful ability to perceive depth, right? So we can tell how far things are. We can tell that things have three-dimensional structure. We can tell that they have depth, right, because of this depth perception, because we have binocular vision. We have both eyes working together here that allow us to have this better sense of vision. So if you were to damage one eye, right, if you were to damage one eye or you damage an optic nerve or something, you're going to now lack that depth perception because you'd only have one eye functioning instead of two, okay? So you have to have both eyes, this binocular vision, in order to have proper depth perception, okay? So that is the end of our eye lecture. We covered a lot of material, so please review it carefully. And always, if you have a question, if you have any concerns about any information that we discussed, if something doesn't make sense, or you just need some clarification, please feel free to reach out. All right, I will see you in the next video.